Deep within the cell's nucleus, there's our DNA. DNA is made up of genes, and each gene is basically a specific part of the DNA that codes for a protein. And genes become proteins in two steps: transcription and translation. Transcription is the first step in creating a protein, during which a specific gene is read and copied on an individual mRNA, or messenger RNA molecule, which is like a blueprint with instructions on what protein to build. Now, DNA has two strands, which wrap around each other to form the characteristic double helix. Each single strand of DNA is composed of 4 types of nucleotides, which are the individual letters or building blocks of DNA. Nucleotides of DNA are made of a sugar, deoxyribose, a phosphate, and one of the 4 nucleobases, adenine, cytosine, guanine, and thymine, or commonly ACGT for short. The nucleotides on one strand pair up through hydrogen bonds with nucleotides on the opposing strand to create a double-stranded DNA. Specifically, A bonds with T and C bonds with G, so they're called complementary bases. Now, with these two strands, one strand is called the coding, or the sense strand, and the other strand is called the template, or the antisense strand. The coding strand has a coding sequence of nucleotides that serves as a master blueprint for our protein. It's a what you see is what you get kind of thing. The template strand, on the other hand, has a sequence of nucleotides that's complementary to the sequence on the coding strand. In addition, the two DNA strands also have a direction. The coding strand runs from the 5' end toward the 3' end, while the template strand runs from the 3' to the 5' end. Kind of like two snakes coiled up together but facing different directions. So if the coding strand looks like this, 5' end, AATCCAGTA, 3' end, the template strand would look like this: 3' end, TTAGGTCAT, 5' end. Now, transcription starts with the unpacking of DNA from chromatin and dehelicization, meaning that the double helix unwinds a bit so that the individual genes are exposed. The starting point of a gene is determined by a promoter region, which is a repetitive, non-coding sequence of nucleotides. For example, TA sequence is one super famous promoter, called the Tata box, and it marks where to start transcribing. A few dozen proteins and enzymes come together to form what's called a pre-initiation complex around the promoter, also featuring an enzyme called RNA polymerase. Then a process called elongation happens, which is where RNA polymerase unzips the two strands by shearing the hydrogen bonds between the complementary nucleotides for the length of around 14 base pairs. This open area is within the RNA polymerase and is called the transcription bubble. The RNA polymerase follows the template strand and uses it to assemble an mRNA molecule, which is a mirrored image of the template strand. Now, mRNA is slightly different from DNA. First off, it uses a slightly different set of nucleotides, where the T is replaced by uracil, or U. The U will normally pair with A, as T would. Also, mRNA runs in the opposite direction compared to the template strand, so from the 5' end to the 3' end. So when reading the template strand, RNA polymerase will move along from the 3' end of the template strand towards the 5' end, while creating the mRNA molecule in reverse, from 5' end to the 3' end. RNA polymerase assembles mRNA by attaching complementary nucleotides. For example, if we're making a protein based on our coding strand from before, which is 5' end, AATCCAGTA, 3' end, the template strand looks like this: 3' end, TTAGGTCAT, 5' end. And the mRNA will look like this: 5' end, AAUCCAGUA, 3' end. A striking similarity. As RNA polymerase goes down the DNA, the hydrogen bonds between the nucleotides that have already been transcribed reform, zipping the DNA back together. Eventually, RNA polymerase reaches a specific sequence called the terminator sequence. The terminator sequence contains two complementary sequences in a row, on a single strand. When they get transcribed into the mRNA, the newly formed sequences bond with each other, creating a hairpin loop. And that makes the RNA polymerase detach from the DNA strand by simply yanking it off. That causes termination of transcription. mRNA detaches from the enzyme, DNA closes back up, and RNA polymerase goes off to transcribe another gene. But the mRNA is not done yet, and requires some modifying. For example, a 7-methylguanosine cap is added at the 5' end of RNA, and at its 3' end, the enzyme polyadenylate polymerase adds a polyadenine tail, meaning a long sequence of only adenine nucleotides. Think of these as those plastic bits on shoelace ends. Their purpose is to make the mRNA more sturdy. But the story isn't over, because the mRNA has to undergo some editing. mRNA can be divided into sections of exons and introns. The exons code for the protein, while the introns don't. So to trim down the unnecessary bits, a molecular machine called the spliceosome comes in and splices the introns out. This is kind of like video editing, when you cut material from the initial filming to make a much cleaner final cut. In the end, we get a sequence of exons only, with a cap and a polyadenine tail, which is ready to code for a protein. The freshly made mRNA floats out of the nucleus and hooks up with an idle ribosome in the cytoplasm, where it can create a protein. Alright, as a quick recap. The DNA is made out of genes, which can be converted into mRNA protein blueprints through a process of transcription. The promoter is the beginning of the gene. The RNA polymerase binds to it and starts assembling mRNA from it. In the end, the mRNA detaches and proceeds outside of the nucleus.